the four of us. Yeah, and is it okay if we record this meeting?
Sure. Yeah, go ahead.
All right, thank you. So I'll just start off by introducing us and then introducing our project to you. It shouldn't take too long, 10 to 15 minutes. And then we can have a discussion. Like, you can feel free to ask us any questions or give us any tips or advice about any like holes you may have found in our project. So yeah, I'm Anya, you know me, and then we have Hannah, Audrey, and Aishani, and they'll just be here helping and answering questions with me. So to start off, our project is called Vigilantly Optimizing Cancer Detection. We kind of tied in VOC into it with the Vigilantly Optimizing Cancer, and it's our project for the 2021 season. Like I mentioned to you earlier, this is a synthetic biology competition, so we have to create sort of a solution to a certain problem using synthetic biology. So next slide. Hannah. Oh, thank you. Okay, so just a little statistics that you're probably familiar with. The problem is that lung cancer is the second most common cancer and the most fatal cancer in the world, accounting for the most casualties annually. And the lack of awareness leads to 7 out of 8 people not getting the recommended annual screening. The rate of lung cancer has increased over the past 3 decades, with most cases detected late after stage 2. And here are some statistics. So there's 131,880 lives lost annually, and 21% of patients have a 5-year survival rate, and 25% of cancer-related deaths are related to lung cancer. So we took this problem and we realized that the big sort of root cause was the late detection. So and then we found that VOCs, or volatile organic compounds, are biomarkers for lung cancer that are present in the human breath. So these compounds have a high vapor pressure and low water solubility and tend to be present in higher concentrations in lung cancer patients. So they can potentially be used to create like a non-invasive detection system or detection tool that can help with early detection of lung cancer. So we took this information and we created a solution. So our plan is to create a product that allows for early detection through a breath test that has this detection system. So more specifically, we'll be using E. coli bacteria to detect the VOCs that we identified to be associated with lung cancer. And then if E. coli detects these VOCs, a regulatory promoter, which we recently, just like less than a week ago, decided to do a transcription factor instead of a promoter, because after extensive research, we found that a promoter related to a VOC that could help with this detection system kind of just did not exist or did not work. So we will use a transcription factor, which we are currently researching right now. So we hope that this will detect the presence of lung cancer in the patient and express like sort of green fluorescent protein or something that allows for scientists or medical professionals to see if the biomarker or VOC or biomarker or lung cancer is present in the patient. So this is a visual representation of our previous plan, but our plan was basically to take E. coli and detect if the VOCs were present, and if they weren't
Anya,
present. Yes.
Sorry, can you go to the previous slide?
Yeah.
Okay. Sorry, I was unable to follow through the slide. I'm so sorry. Can you just repeat on this slide, please?
Yeah, of course. So I was a bit confusing, sorry, because we just changed our plan. But our previous plan was to take E. coli bacteria that will detect the VOCs or the specific VOCs, and that will then sort of turn on a regulatory promoter, which will then like express a reporter gene. So we were supposed, like, it's kind of like a flow chart where we would have the E. coli, which would detect it, turn on the promoter, which would then express the reporter gene, and the reporter gene would then let the medical professional know that this is present in the patient and they thus have lung cancer. But recently, we like the whole promoter thing was hard for us to find because we have to create a model of this, and we changed it to a transcription factor. So the details on that are a little blurry for me right now. If a team member wants to elaborate, that would be. Yeah, I think we're still researching on that. So does that a little bit clearer for you or?
Yeah, so basically I'm trying to understand, are you planning to use E. coli to express the VOC? Or is that the question here?
So our plan is to use E. coli to help us detect the VOC.
And how do you do that?
I think
How do E. coli can detect VOC?
Hannah, do you want to elaborate on that?
Yeah, so our current plan, we shifted a little bit away from what is on the slide. So our current plan is to find transcription factors that bind to these volatile organic compounds. And then these transcription factors can either like upregulate or downregulate a reporter gene like GFP, for example. If you'd like me to go a little bit more in depth, that's just like the 1 second summary.
I'm trying to, so basically you're trying to say the transcription factor which regulates GFP has the capability of binding to the VOC.
Yeah, so we're trying to engineer some sort of system, some sort of sequence, which has some sort of reporter gene as well as a promoter that is a promoter or transcription factor that is dependent on a VOC.
Okay, and do you guys know anything about specificity or available combination of VOC and transcription factor, or it is just an hypothesis?
Right now, we're trying to research and find some sort of transcription factor that is specific to VOCs. When it comes to, yeah.
And did we talk about the VOCs, like what exactly these organic compounds are? Sorry if I missed on the earlier slide.
Yes, but we didn't go into I didn't go into like the specific like VOCs themselves, like propanol, octane, and etc.
Got it. Now I got it. Okay, go ahead. Sorry, go ahead. Yeah.
No, thank you for okay. We can just probably skip to like yeah, we can probably skip through this. So this was just a flow chart that basically explained it just more visually. And this was our previous plan, but if VOCs were present, the promoter would be activated, reporter gene would be expressed, and then it would be detected. So, but Hannah explained it really well.
I got it. Okay.
So along with that, we wanted to go a little bit into the software side where we wanted to create a machine learning algorithm that would predict asthma levels using VOCs. So you might be wondering like why asthma? So we wanted to quantify the expression of the reporter gene and compare it to normal VOC levels, but then like in asthma, there are VOCs still present. So we need to be able to distinguish between asthma and cancer as well, if that makes sense. So we also plan to create molecular modeling of the VOC binding to the transcription factor, which would then indicate if lung cancer is present. And then, so another aspect of iGEM is science communication, which is what we're actually doing right now. We collaborate with other teams, like we have the CCA iGEM team that we collaborate with weekly, and Westview iGEM team. And we also have workshops. We actually had workshops which we recorded and uploaded to YouTube, and we did those with local middle schools on synthetic biology and lung cancer. And we interview experts like you, doctors, oncologists, specialists, etc. Anyone who can help us and guide us in the best way possible, like you're doing right now. And we're really grateful. And next slide. And last but not least, there's human practices, which is just spreading awareness of our project online or through the community. We post infographics on social media, and we create surveys for community engagement just to gather data, see how many people are aware of our project, lung cancer, and how much we can, and then we record how much we contributed to the community with our project, through outreach, and human practices is also centered around additional interviews with previous patients or other things. And that's our project. So any questions at all, you can just go ahead.
Okay, so that's all the slides you all have?
Mhm.
Okay, great. So what is the exact end goal? Are you guys like trying to develop a project for, what is the application of this? Or it is just or it's just an information gathering and, trying to develop a kind of information like, how do you say, like a library of information on the hypothesis that you guys have?
So I think my team members can elaborate on this if I'm wrong, but with iGEM, we just need to have like a plausible sort of solution, like a hypothetical solution rather than like a physical solution.
Okay.
Like in real, we can't go into labs currently
Sure.
because of COVID, so
Yeah.
we can't actually create something, but we should have like all the information, like you said, ready to create something.
Right. Fantastic. First of all, I'm really apologize. Like I was like 5-6 minutes late. It doesn't look, it looks bad on me, but I'm glad we connected, and I'm glad to help you guys, to advance this program, or to have the information you're seeking. And just a little background, Anya maybe a little bit knowing, but for the team, I have been involved in a lot of oncology drug development, and have worked with some of the big cancer hospitals in US, like MD Anderson Cancer Center, in Houston, and also at National Cancer Institute in Bethesda, Maryland. And currently, like, I'm the director of drug discovery in a biotech company, and I'm glad to help your young team towards this information you're seeking. So the first question is, tell me more about the VOC. What exactly you are looking when you say VOC? Or if it is like, something confidential, you guys don't want to share, it's perfectly fine.
It's not confidential, but does anybody else want to answer? Or
I can jump in here. So we gave a bit of a technical definition in our presentation, but a VOC is pretty much any compound with a high vapor pressure, so it vaporizes easily in air. So in the context of lung cancer, there's been a good amount of work done linking various concentrations of VOCs, whether it's higher or lower, to lung cancer. So let's say, whereas a lung cancer patient would have probably like 5 ppb of propanol, for example, maybe a healthy patient would have 0.1. So we pretty much based our research off of that. So by consulting a database of VOCs whose levels were changed in lung cancer patients versus in healthy patients, we were able to kind of build off of those list of VOCs in order to develop the promoters and transcription factors for our own project.
yeah, and adding.
Sorry, go ahead.
Sorry. So just adding on to that, VOCs are just present everywhere. Like, this is not really relevant to our project, but like paint actually emits VOCs because they're sort of like more toxic compounds. So
Sure.
that's why they're more present in like.
Yeah. I understand. Like, to any kind of inflammation in the body, whether it is, as you were talking about asthma, right? Asthma. Or even a regular or normal healthy volunteer or donor will have some level of VOCs, depending upon the stress level, right? Like, and these are, and definitely in situations like cancer or asthma, like, which is an inflammation or autoimmunity, there will be a very high amount. And I think that is what I saw, like, you guys are trying to compare, your transcription factor binding to the VOC to a normal donor, binding to asthma level patients, binding to cancer, and then you have this comparative levels of expression, and to compare, and to check. So sorry, Hannah, did you mention a name of VOC?
Yeah, so I just gave an example like a hypothetical example, but we have pinpointed a couple of different VOCs, such as like trimethylbenzene, styrene.
Okay, got it. Okay, yeah, thank you. Yeah, so okay, yeah, quite interesting. And actually, the entire concept of this project to develop biomarkers, right, to identify patients or possible patients well early on the basis of VOC induction, as cancer initiates, right? Like, because, all of us here, we have what we call, a very small, how do you call, like precancerous cells in our body. But there are different events, genetic events or mutations which happen, which amplifies those signals, right? And when those signals are amplified, obviously, VOC can be one of the biomarkers, looked into. So, development of biomarker is I'm just giving a little background about the because I also work on lung cancer, significantly on lung cancers, on melanoma, skin cancer, and companies have been developing various drugs, right, to treat cancer, but drugs have not been 100% successful, and this because right drug doesn't go to the right patients, or the right drug doesn't go to the patient well early because the patient has been not detected. And as you mentioned correctly, most of the cancers are detected in stage 3 and stage 4 when you have some kind of symptoms, right? You're not just going to go for a cancer detection like today, like, oh, can unless you show some symptoms. So this kind of biomarkers, like early biomarkers, and and you guys are working on VOC, that like there are multiple other things, which I'm also working on biomarker development for identification of this clinical or these patients, in early stages. So it's and identifying this kind of biomarkers or such biomarker will not only increase the percent efficacy in clinic, right? For example, today drug A is used for treating lung cancer, and out of 100, only 20 people, their survival is extended, like you saw, right? Like you presented in the first slide, the survival rate is 2 to 5 years, right, in lung cancer patient. But by giving drug A, out of 100 patients, if 20 patients, you can extend the life from 5 to 10 years, that's, you're enhancing this, but it's only achievable in 20%. What happens to those 80%? Why didn't those 80% responded, right? Maybe they were at a very late stage of cancer development, and that is where your program fits into, and maybe identify like well early that out of 100, only this 20%, only these 20 patients can respond to a treatment. So instead of those selecting, you can exclude those 80 people or patients, and you can only do the clinical trial on this 20. Now your success rate is 100, right? Because you're treating 20 out of 20 patients. But you can identify biomarkers, similar biomarkers for other 80 patients, right? They may have some other biomarkers in their system. So that is where biomarker plays a very significant role, and down the line, I can share some very general reading to you guys, which you can review and can build your like introduction, or like you can write a kind of a summary or project, something which I can help you to write up or review, like aim, hypothesis, and what is the the experimental part, how you can do it, because you guys are not going to do in the lab, right, because of the COVID, but you can develop a project right from what is the question. So like the background, that Anya gave, right? There was a little background, right? Lung cancer is the second deadly cancers, and I can actually give you guys the exact numbers, what's there happening, and and what are the different treatments for the lung cancer patient, and how does biomarker play a role. And once you have developed all those questions and hypotheses, and the experimental part is where you will talk more about how you plan to detect this biomarker. And this is your question, right? Like VOC, if you have a transcription factor, or something which can bind to VOC, and at the same time can bind to the promoter of a gene that can be expressed, which can be measured by some machine, right? Like quantified, and you can detect. So all those can go in the experimentation part, and definitely you can leave, the results. In this case, just to give some directionality, for example, lung cancer, and we know, right, like smoking is one of the major causes or enhances, the chances of lung cancer incidence. And when there is smoking, and just like, some of these chemicals you mentioned, Hannah, different chemicals, when smoking is there, it's always related to some kind of VOC. I'm 110% sure if you detect smoking induced VOC in lung cancer, maybe you will find a lot of things, which can be used. And now we have to see if there are systems where we can pick up any of these very significant or important VOC, which can bind to some transcription factor where we can, binding to, like, the promoter region of some gene we are trying to express. So yes, it's a good project, and the most important thing is in it, the question. Like, if the question doesn't have a solid background, right? If it doesn't have a solid platform, the project cannot be strong. So if we can highlight I can share a lot of information with with your team, and if you can highlight why biomarkers are important, right, today in early cancer detection, how it can impact, increasing the efficacy or survival of patients. And this is your plan, right? And then you can give a directionality, right? Mutagenesis, right? People working in, like, there are hazard occupations, right? Like scientists, they're working in, with reactors or radioactive substance, or smoking induced or carcinogen, like, induced lung cancer. And always they will have some kind of metabolites in their system that you guys are calling VOC, volatile organic compound. If there will be definitely a lot of information that we can pull out, and we can tie it to some kind of molecular biology study or gene expression study with a readout.
Yeah, definitely. You helped a lot with giving us more background. So thank you so much. And it really helps when we have a clear background and show how big the problem is, that our solution seems all the more effective. So thank you so much. And we'll definitely run things by you if we have any more questions on that. But do any of you have questions? Like, any team members have any questions that you want to ask? Okay, then I'll just ask. So what problems or risks might you foresee with this kind of diagnostic test, like with VOCs? Is there anything that you think we should be concerned about or should address?
The thing is, because the way I do biomarker analysis is very different. We do a lot of gene sequencing, right? And I don't know if VOCs are related to kind of genetic mutations. Like, if there is a genetic mutation which predisposes a person to cancer, lung cancer, right? And if VOC is the mediator of that genetic mutation, if you all can find something like that, right, that will be an added advantage. So I haven't worked with a lot of VOCs because I have a different profile. I look into different area of biomarkers. But when I generally think, to determine, because if these are just organic volatile, we have to tie it to something. We just cannot take. There may be so many organic compounds or volatile organic compounds, as you're saying, which will be present, like in a cancer patient, right? We have to identify which are basically more related to carcinogens. Like, they're known to be carcinogen. They are related to the cancer pathway, cancer signaling pathways, right? Because a VOC in asthma may not cause cancer, right? A VOC in a normal person may not be a normal person. When you take, right, and you expose that person to any kind of stress, like physical environmental stress, right? You may have different kind of metabolites. Are the same metabolites expressed in cancers? So how you are going to use that as a biomarker, right? So you have to really filter it down to very specific VOCs, which are highly related to cancer initiation and cancer progression, right? So what you have to avoid, what I see the challenges are, is identification of VOCs which are specific to cancer initiation and disease progression. Otherwise, you may pick up a non-specific background and you may keep spending time with it when that VOC may be, present in other stress condition too. So that is something, and definitely, how sensitive. Other challenges would be, sample collection, right? What kind of sample collection? Second is how sensitive your instrument would be to measure or capture that, right? And obviously, the molecular biology challenges. Do you have, gene expression systems where your VOC can bind to transcription factors or promoter regions of of these transcription factors? Because what you're talking is a compound which is volatile, right? It is not stable. So at what time of do you try to identify, capture these VOCs? Are these VOCs very high during the first month of cancer initiation? That is, just at the start when the cancer is starting. Do you miss that window, or it is the second to fifth month, right? So what time frame of cancer initiation and disease progression your VOC will be highly detected, or makes a sense to capture them, right?
Right. Okay, that makes sense. I think we need to look more into the timing because we haven't really looked into that, and
Sure.
specificity too. And then thinking, so based on your experiences with various different cases and everything, do you think that there are any ethical concerns regarding our diagnostic tools, like bioethical concerns or anything that we should look out for?
Yes. So any kind of diagnostics when you develop, it has to be approved. It has to be like FDA approved. And mostly what the clinicians, definitely the patients, it's the primary call by the patients, right? Whether they want to get, first of all done, right? And second thing is whether the clinicians are okay doing that, using the diagnostic tools. How easy it is to operate. For example, you cannot just go and ask someone to give a tumor sample, right? A cancer patient, because if it's a solid tumor, that means you're taking a biopsy, right? Which is painful, time consuming, and needs a surgery or operation, right? Can your diagnostic tool be very simple, like just drawing a blood, right? And if you can identify, or in your case, like if it is like the breath, right? That is what we're talking, right? Uh, um, does that is a good biological sample, right? And also, yeah, I think how easy it is to collect those biological samples, right? If there is something where some kind of instrument where you can just take a swab in the nostril, if it works, it's good. That will be preferred by a patient and even a doctor, rather than taking the cancer, like the initial cancer lesions, if they're appearing somewhere. So yes, how you collect the sample is ethical concern, and it need to be FDA approved. And definitely it should not be invasive. It should not be invasive. It has to be as simple, as easy as possible, both for the patient and the clinician.
That's it for me. Does anything else to add on? Any follow-up questions for any team members? Do you, Mr. Rajiv, do you have any adding on feedback or any last questions for us before we wrap up here?
No, but feel free if you guys want, any kind of more literature or write up, or if you like to, share like, a project, like if you guys are writing up something, right? And if you want a structure, like how it should be written, if it is to be reviewed, or edited, or maybe there are so many technologies that, definitely you are not aware, right? So I can pitch in those technologies, or instrumentations which can be used for creating, like, there is, so many, high throughput instruments right nowadays, people use as a diagnostic, machine to capture these signals, and also a lot of background information. So, feel free to, to reach out to me, anytime, any of you, if like to discuss more, get more information, writing, reviewing, or any other scientific projects you have.
Yeah, thank you. We really appreciate it, and we'll definitely let you know because we're getting to that stage of our project. So, really helps. So, thank
Yeah,
you so
sure.
much. Thank you so much for meeting with us today and giving us your time.
Hey, no problem. Always, and it's always exciting to see, the young team, who have interest in all these, very complicated cancer detection and therapeutics. It's really exciting to see, the younger generation getting involved and having interest, putting time. So glad if I'm of any help to you all.
Thank you.
Okay.
Thank you so much.
Okay.
Thank you.
Thank you. Have a good one.
You too.